today's video we are going to learn about the evolution of the parenteral preparations. The finished parenteral products are subjected to various evolution tests in order to maintain the quality control. What are those tests means? The first test is the sterility test, clarity test, leakage test, pyrogen test and assay. In order to evaluate the parentrals, we have to conduct all these tests. So let us learn about the first test, that is the sterility test. All the parenteral preparations which are supplied in the sterile form must conform to the test for sterility as prescribed in the pharmacopoeia. The test for sterility is done by detecting the presence of viable forms of bacteria, fungi and yeast in the parenteral preparation. The sterility test must be carried out under strict aseptic conditions in order to avoid the accidental contamination of the product during the test. Here all the glass waste required for the test must be sterile. Culture media required for the growth of the aerobic and the anaerobic and fungi are prepared as described in the pharmacopoeia. The test for sterility may be carried out either by membrane filtration method or by direct inoculation method. So the test samples of the parenteral preparations are transferred into a test tubes containing a sterile culture media for aerobic, anaerobic bacteria and fungi. These test tubes are incubated for a stated time in a incubator. The presence of the turbidity in the culture media indicates the growth of the microorganisms and the sample fails to comply with the test for sterility. So this can be confirmed by repeating the test. So this is how we will have to conduct the sterility test for the parenteral products. We will see clearly. So the first method is the membrane filtration method. The method is preferred for oily preparations, ointments that can be put into a solution, or non-bacteriostatic solids not readily soluble in the culture media, or for soluble powders or liquids that possess a bacteriostatic and a fungistatic properties. So for this type of preparations we have to prefer membrane filtration method. This method involves the filtration of the sample under test through a filter media having a normal porosity of 0.45 microns and a diameter of approximately 47 millimeters. So prefer the filter media measurements like that. After that we have to conduct the filtration process. So after the filtration, the membrane is removed aseptically from the metallic holder and divided into two halves. Okay. After that, the first half is transferred into a 100 ml of culture media meant for fungi and incubated at 20 to 25 degrees centigrade for not less than 7 days. So we already know the incubation period for any type of culture media is 7 days. The other half is transferred into 100 ml of fluid thioglycolate media and incubated at 30 to 35 degrees for not less than 7 days. Okay. After 7 days we have to observe the growth in the media. So here is the formula for the fluid thioglycolate media. So how to prepare this media means, so mix all the ingredients except the thioglycolate and the residurin in a mortar and stir in some hot water, transferred into a flask and add the remaining water. So make the clear solution by heating in a boiling water bath. Add the sodium thioglycolate, then 1 normal sodium thioglycolate so that the pH of the media will be 7. So reheat the solution. After that filter and add the residurin solution. So transfer into a final container, sterilize by autoclave for 20 minutes at 121 degrees centigrade. Cool properly to 25 degrees centigrade and store at 20 to 30 degrees centigrade, avoiding the excessive light. So media more than 3 weeks old should not be used. So during storage, if more than 30% of the upper portion of the media has changed to a pinkish color, it is unsuitable for use. Okay. So prepare a media, culture media like this. And the next method is the direct inoculation method. In this method, the specified quantity of the sample under test is drawn aseptically from the container and transferred into a vessel of the culture media. Mix the fluid with the media and incubate for not less than 14 days here. So observe the growth of the microorganisms in the medium. Observation and the results. The culture media is examined during and at the end of the incubation to find out if there is any microbial growth. So the following observations are possible here. What are those means? The first one is if there is no evidence of the growth, hence the preparation being examined passes the test for sterility. Okay, if there is the evidence of a growth, what we will do? So retesting is preferred using the same number of the samples, volume to be tested and media as in the original test here. So that is about the second situation. So what is about the third situation? So if there is evidence of a microbial growth, so isolate and identify the organism. If they are not readily distinguishable from those growing in the container reserved in the first test, so the preparation being examined fails the test for sterility. They are readily distinguishable from those growing in the containers reserved in the first test. The second retest is preferred using twice the number of the samples. So the preparation being examined passes the test for sterility in case there is no evidence of the microbial growth. In case there is evidence of growth of any microorganisms in the second retest, so the preparation being examined fails the test for the sterility. So this is how we will calculate the results. The next test is clarity test. Clarity test is performed to ensure that the parenteral products are free from foreign particles. So each parenteral preparation in its final container is subjected individually to a visual inspection to exclude the possibility of the foreign particles. The unlabeled containers are held by neck against a strong illuminated black and a white screens. Here white screen is used for the dark colored particles and black screen is for detection of the light colored particles. The contents of the containers are slowly inverted and rotated. The solution is examined for the presence of the foreign particles. If any foreign matter is visible, the same container is rejected. Nowadays, various instrumental methods such as Coulter counter has been developed, which are widely used in industries. So all the parenteral products must be sterile. They should be free from parabens and particulate matters. So the particulate matter is mainly defined as unwanted mobile insoluble matter other than gas bubbles present in the given product. So the presence of the foreign matter in the parenteral preparation is very dangerous, especially when its particle size is larger than the size of the RBC. So it can block the blood vessels. So the parenteral preparation showing the evidence of the contamination with the visible foreign material should be rejected here. The permitted limits of the particulate matters as prescribed in IP is given in the table here. Here the particle size in micrometer, the maximum number of the particles per ml is given in this table here. See here. So what are the sources of the particulate matter means? Here the particulate matter may enter into the parenteral product by two means of sources. What are those means? Intrinsic contamination and extrinsic contamination. So what is intrinsic contamination first means? So the material which are originally present in the parenteral solution are known as intrinsic contamination. Ions leach in the parenteral product which may react with the sulfur ions in the product to form a barium sulfate crystals. So this is about the intrinsic contamination. So what is extrinsic contamination means? So the materials which comes from the environment and contaminate the parenteral product are known as extrinsic contamination. For example, shedding of the material from the body and clothes of the persons, ceiling, wall and furniture of the room. So the particulate matters includes materials like cotton, glass, rubber, plastic, tissues, insect fragments, dust, paper fragments, bacterial contaminations, etc. So this is about the extrinsic contamination. So by these two sources of contamination, the product may get contaminated. So here what are the methods for monitoring the particulate matter contamination? There are mainly four types of methods are there. for to monitor the particulate matter contamination. What are those means? The first one is visual method, counter method, filtration method, and light blockage method. First method is the visual method. It is an old but reliable method. Here, according to this method, the filled containers are examined against a strong illuminated screens by holding the neck and rotating it slowly or inverted it to exclude the possibility of the foreign particles. So if any particulate matter is visible, that container is rejected. So that is about the visual method. And the second one is the counter method. So the method is based on the principle that increases in the resistance is observed between the two electrodes as the particle approaches and passes through the orifice. An electrolyte is required to be included in the preparation before its evaluation. The particles with diameter below 0.1 micrometers can be detected by this method. So it is somewhat high accurate method nowadays. And the next method is the filtration method. So this method is meant for counting the particles in hydraulic fluids. The liquid sample is passed through a filter and the material collected on the surface of the filter is examined under microscope. The method requires skilled and high trained technicians here. And next type of method is the light blockage method. The method gives an automatic evolution of the particles in a hydraulic coils. It allows a stream of the fluid under test to pass through a bright white light source and photodiode sensor. It is possible to detect the cross-sectional area of the particles in this instrument because it blocks the path of the light, and size of the particle is considered as diameter of the circle of the equivalent area. So this is another type of method. The next test is leakage test. This test is preferred only for ampoules which have been sealed by fusion to ensure that there should not be any leakage in them. Leakage test is performed in a vacuum chamber. Here, the ampoules are dipped in 1% solution of methylene blue in vacuum chamber and vacuum is applied here. When vacuum is released, the colored solution will enter the ampoules with defective sealing. The presence of the dye in the ampoule confirms the leakage and hence rejected here. Vials and bottles are not subjected to this test because of the flexibility of the rubber. So this is about the leakage test. Pyrazine test is done to check the presence or absence of the pyrazines in all aqueous parenteral preparations. So what are these pyrazines means? So pyrazines are the metabolic products of the microorganisms and are produced by all microorganisms. But gram-negative bacteria produce most potent pyrazinic substances. Pyrazines are the polysaccharides and are thermostable in nature. They are soluble in water and can pass through bacteria-proof filter. They are unaffected by bactericides also. So we can't kill these pyrazines. So the final option we have here is to prepare a product which should be free from pyrazine. Okay. And principle. So the test involves the measurement of the rise in the body temperature of the rabbit following intravenous injection of a sterile solution of a substance being examined. Here rabbits are used to perform this test because their body temperature increases when pyrazines are introduced into their body by parenteral route. Here materials required for the test are the temperature recording devices, glassware, syringes, needles. Treat all diluents and solutions for washing and rinsing of the devices in a manner that will ensure that they are sterile and pyrazine free. The retaining boxes for the rabbit in which temperature is being measured by electrical devices should be made in such a way that the animals are retained only by loosely fitting neck stock and rest of the body remains relatively free so that the rabbit may sit in a normal position. So the animal must be put in the box 1 hour before the test and remain in them throughout the test. So ensure that the room temperature where the test is carried out is within the range of 3 degrees centigrade, or the place where the rabbit have been kept for at least 18 hours before the test. Or withhold the food from the animal overnight and until the test is completed, and also withhold the water during the test. So here we will pick up 3 healthy adult rabbits of either sexes, each weighing not less than 1.5 kg, fed on complete and balanced diet and not showing any loss of body weight during the week preceding the test. So do not use any rabbit which is having a temperature higher than 38.8 degrees centigrade and also showing a temperature variation greater than 0.2 degrees centigrade between 2 successive readings in the determination of the initial temperatures. So coming to the method how to conduct this test means, here the test is carried out in an air conditioner room. Dissolve the substance being examined in or dilute with a pyrazine free saline solution. Warm the liquid being examined to appropriate 38.5 degrees before injection. The amount of the sample to be injected varies according to the preparation being examined and is prescribed in the individual monographs. So the volume of the injection is not less than 0.5 ml per case and not more than 10 ml per case of the body weight. Withhold water during the test. Clinical thermometer or the temperature sensing probe is inserted into the rectum of the rabbit for recording the body temperatures. Here 2 normal readings of the rectal temperature should be taken prior to the test injection at an interval of half an hour, and its mean is calculated, which is the initial temperature of the rabbit here. Okay, and the solution under test is injected slowly through an ear vein in a volume of 0.5 to 10 ml per case of the body weight. Record the temperature of each rabbit in an interval of 30 minutes for 3 hours after injection, and the difference between the initial temperature and the maximum temperature recorded for a rabbit is taken to be its response here. So next is interpretation of the results. If the sum of the response of the group of the 3 rabbits does not exceed 1.4 degrees centigrade, and if the response of an individual rabbit is less than 0.6 degrees centigrade, the preparation being examined passes the test here. If the response of any rabbit is 0.6 degrees centigrade or more, or if the sum of the response of the 3 rabbits exceeds 1.4 degrees, continue the test using 5 other rabbits. Okay, so if not more than 3 of the 8 rabbits shows individual response of 0.6 degrees or more, and if the sum of the response of the group of the 8 rabbits does not exceed 3.7 degrees, the preparation being examined passes the test here. Okay, this is how we will calculate the results. And the next test is the assay. So assay is performed according to the method given in the monographs of the parenteral preparations in the pharmacopoeia. So assay is done to check the quantity of the medicament present in the parenteral preparation. So according to the type of the medicament and the category of the medicament, we will conduct the assay as per the monographs of the IP. So this is all about the evolution of the parenteral preparations. Thank you for watching this video. Please subscribe to our channel for more videos.